Hello everyone and welcome to our first lecture within the cell biology lecture series. Today we are going to be talking about cell compartments and also cell organelles. So what is cell biology and why should you care? Well, you're going to become pharmacists, not biologists. You need to know cell biology because it is the study of cell structure and function. And it revolves around the concept that the cell is the fundamental unit of life. Most importantly, it permits a detailed understanding of the tissues and organs that cells compose. So, again, why is it relevant? It is relevant because insufficient understanding of pathological and therapeutic mechanisms at cellular level contributes to the growing difficulty of bringing new drugs to the market. So most of you might know that there is currently no working and efficient drug against dementia, which is common in Alzheimer's disease. And one of the reasons is because our understanding of the cellular processes contributing to the death of neurons or nerve cells in the brain in Alzheimer's is not there yet. We don't understand this in total. Also, cell biology is crucial to understand biochemical and biological aspects of the action of drug. In the following few weeks, Dr. Cottrell will teach you a lot about biochemistry. I'm teaching you about the genetic background of the drugs. However, all of this needs to be combined, and this is basically what cell biology is about. It unifies what you will learn in genetics, biochemistry, and will lead to a deeper understanding of physiology, but also deeper understanding of the action of drugs and possibly also the side effects of drugs. So it is essential in order to understand how certain tissues react to drug. If a pharmacist does not know the basics of cell biology, he would not be able to understand potential side effects of the drugs at cellular and systemic level in the human body. So students frequently ask me the following question: what books do I recommend as a further reading regarding the content of both the genetics lecture series and also the cell biology content? I personally love the book Molecular Biology of the Cell, published by Alberts and colleagues. The edition doesn't play a major role. You could use the 6th or 5th edition. They are both excellent, and this book is also present in the library, so you can basically borrow this book. Please have a look. I personally really like it. In addition to those books, I also do recommend Wikipedia. This might be different to things your teachers told you at school, but Wikipedia is actually not that bad, at least if it comes to basics of certain mechanisms. So I wouldn't rely solely on Wikipedia. It's all about the combination of sources. You can easily check things on Wikipedia out to get a level of basic understanding, and then supplement this knowledge with knowledge from textbooks. This is perfectly fine. So what are the learning outcomes for the session today? By the end of the session, after you watched all 3 videos, you should be able to give examples of approximate dimensions of biological structures in micrometers and nanometers. So please do make sure that you still remember how those units work. If you don't, please go back, have a look at your school books from the school. This is fairly straightforward. And again, if you struggle with those units, go on Wikipedia. The dedicated page is wonderful and really simple. Also, after this session, you should be able to identify the different organelles of the cell and name their functions. Students should also be able to explain why eukaryotic cells are divided into compartments, and also understand the cell as a basic unit of life, a robust system that is made up of really fragile parts. Last but not least, you should know the difference between animal and bacterial cells, and you should be able to explain the details of those differences. Sounds simple, and I think this lecture is quite enjoyable and it's not particularly a challenging one. So let's start. So what you can see here are examples of different biological structures, and what you also see here is how large or small those particular biological objects are. So obviously a hen's egg is a cell per se as well, and it is actually quite large. It's approximately 65 millimeters. However, a typical plant or human cell is much larger. In plants, cells can be between 10 and 100 micrometers approximately. In humans, we have plenty of small cells, but we also have larger cells. However, they are certainly much smaller than a hen's egg. For example, nerve cells in your brain or a neuron has the approximate size of 70 micrometers. However, there are neurons in your body that are significantly larger because your brain is actually connected to the periphery. There are certain neurons that connect your brain to your legs, and they can be up to 1 meter long. And imagine in a giraffe, this is gonna be even more dramatic. Those cells can be several meters long. In contrast to those rather large cells, our body also contains smaller cells, like human red blood cells that lack the nucleus with an approximate size of 7-8 micrometers. So this is all fairly nice, but if we look at bacteria or even viruses, then we need to go down with the dimensions even more. A typical size of a bacterium, for example E. coli, which is normal non-pathogenic bacteria in our guts, is round about 1 micrometer small and 5 micrometer long. So this is significantly smaller than human neurons, and indeed this is actually pretty close to the resolution barrier where we would need to use an electron microscope to be able to visualize it. However, E. coli can still be seen, and this is something you will see in our online practical dedicated to microbiology. So bacteria are quite small. How about viruses? Viruses are even smaller. For example, T4 bacteriophage is only 222 nanometer long, so this is quite small. Human viruses like HIV are in the similar range, with the AIDS virus being just 100 nanometer small. Polio is even smaller with 30 nanometer. So as you can see here, those structures are pretty small. If you don't remember the units I've been talking about before, namely nano, micro, milli, this table is quite useful if you need to remind yourself about the conversion. So please make sure that you are familiar with these units and that you are able to convert them, because this is gonna be not only relevant in part 1 for several practical sessions, but this is something we could assess you on later on in our exams on practical classes, and you will need them throughout your whole career at the university. Those are basically basics units we are using in science, and I think my cat just agreed. So let's move on. What is the cell theory? Cell theory is actually quite old concept. Robert Hooke in mid 600s observed a sliver of cork under a microscope. Back then those microscopes were much more primitive than they are nowadays. He saw a row of empty boxes, and this is basically the structure you can see here. So it does look like boxes. So remember, this is a slice, so he was only able to do is to see this in 2 dimensions. However, he was aware that those empty boxes are actually 3-dimensional, and he already then coined the term cell. 200 years later, a German physiologist, Theodor Schwann, postulated that all living things are made of cells, and he was basically right. This has been confirmed by another German physiologist, Rudolf Virchow, who postulated that all cells come from the cells. So he observed that this basic unit of biological systems can only be generated based on a pre-existing cell. So if we unify those two theories and all we know, then we can easily say that all living creatures, both simple like viruses and complexes, are made out of one or more cells. And we can also say for sure, that the cell is a structural and functional unit of life. Also, all cells are derived from pre-existing cells. And this holds true for bacteria, our blood cells and neurons. All of those cells, although very diverse in function and size, are coming from pre-existing cells, and this is how cell theory actually includes them. So, what are the common characteristics of all cells independent of the species? The first characteristic is a surrounding membrane. And this is a schematic view on the mammalian cell membrane. In addition to the cytoplasm, we have the cytoplasm, to the cell membrane, we have the cytoplasm, sometimes referred to as cytosol. And we also do have organelles. Those organelles are not necessarily characteristics of all cells, because only mammalian and plant cells possess complex organelles, but predecessors of the structures can also be seen in bacteria. So, there are two basic types of cells. We do have the so-called prokaryotic cells, and all bacteria are prokaryotes. This includes bacteria and archaea. Examples of bacterial cells are spherical cells, also called cocci. We can have rod-shaped cells, bacilli, and we can have spiral bacteria. So those shapes can be very, very different. However, the fundamental unit of organization of prokaryotes is a cell as well. The difference between prokaryotes and eukaryotes is that, in contrast to eukaryotes, we don't have a membrane-bound nucleus. What bacterial cells or prokaryotic cells often have instead is a so-called nucleoid, and this is the region of high DNA concentration. And if bacteria do possess organelles, they are usually not bound by membranes. In contrast, eukaryotic cells, including both human cells and plant cells, and this also includes fungi, protists, and so on, and various cells like blood cells, neurons, or skin cells, do have a nucleus which is bound by a membrane. Actually, after close inspection of this slide, I must admit that one cell here does not have a nucleus. Red blood cells don't have it, and the same holds true for platelets. However, red blood cells and platelets are indeed generated by cells that do have a nucleus. So this is basically the result of cellular evolution. So we can claim that in eukaryotic cells we do have a nucleus bound by a membrane. They also possess many different organelles, and those organelles are usually bound by membrane as well. With this, I would like to end up our first brief section, and I will see you for our next session where we introduce several of cell organelles. Thank you very much for your attention.